Hello, Lana. Welcome to our third lesson. And we want to look at preparation of a temporary slide. So to observe a specimen under the microscope, we need to prepare slides. There are two types of slides. Temporary slide for immediate use during laboratory exercise, and a permanent slide which can be preserved for future use. So let us look at preparation and observation of temporary slides of onion cells under the light microscope. What are the requirements? Microscope, slide, cover slips, scalpel, distilled water, iodine solution, onion bulb, a pair of forceps, dropper, and a mounting needle. Procedure: a piece of epidermis is obtained from a fleshy leaf of an onion bulb and placed on microscope slide, then a drop of water added and covered with cover slide gently to ensure no air bubbles are trapped. Observations are made under both low and medium power objective lens. After that, the same procedure is repeated with another epidermal strip. Instead of water this time, a drop of iodine solution is added to stain the cells to make them clear, or by giving the cells color. So after the drop of iodine has been added, cover slip is introduced on top, and observations are made under both low and medium power objective lens. So the cell wall and the nucleus stain darker than any other part, and then just make a drawing of that. So nucleus, cell wall, cytoplasm, and cell membrane. Most parts of a cell are clear and transparent. Hence, when viewed, so they are not transparent and clear, but when viewed under the microscope, they do not appear distinct. To improve clarity, sections for viewing are treated with solutions containing dyes, that is staining, to make them appear distinct and clear. The sections to be viewed should be very thin to allow light to pass through. These sections should also be kept moist before mounting to avoid drying up, which may destroy the cells. So when we want to observe permanent slide of animals, we require to have a permanent slide of animal cell obtained from chicks, nerve cells, and muscle cells. Remember, we cannot observe the blood because of various issues like HIV, AIDS, and other issues. The slide is mounted on the microscope, and observations are made under both low power and medium power objective lens, and then drawings of the cell are made. Apart from that, comparison between plant and animal cell is done. So a light microscope can be used to estimate the cell size. Remember, most cells are shorter than a millimeter, hence measured in smaller units called the micrometers. Usually 1 millimeter is equivalent to micrometer, and 1 micrometer is equivalent to 1,000 nanometers. Those are important units to know. So how can we estimate the size of an onion epidermal cell? We require microscope, transparent ruler, calibrated in millimeters, and prepared slide of onion epidermal cells. After having these, with a low power objective lens, place a transparent ruler on the stage of the microscope, and then focus so that the millimeter marks on the ruler are seen as dark lines. Then estimation of the diameter of field of view is done by counting the 1 millimeter spaces between the first mark and the last one across the field of view as shown here. For example, the diameter of field above is estimated to be 2.25 millimeter and has to be converted to micrometers. So this is the conversion unit. So 1 millimeter is equivalent to 1,000 micrometers. So that will give us 2250 micrometers. The ruler is removed and the prepared slide of the epidermal onion cell is placed and focused to obtain a clear image. The number of cells along the diameter of field of view are counted, and then the length of cell is calculated using the formula. The cell length, or the length of one cell, is calculated by dividing diameter of field of view in micrometers over cells counted along the field of view. So assuming 6 cells were counted along the diameter, then the length of one cell will be the diameter of field of view in micrometers, which is 22 or 2250 divided by the number of cells counted along the diameter of field of view, and there are 6 of them. So one cell measures 375 micrometers. So we are through for that lesson, but wind up this assignment.